Okay, surprising. So I'm Randy Vita. I'm going to talk about how we get the data retrieved, entered, and organized. So the scope of IEDB, already mentioned, was infectious disease, allergy, autoimmunity. We had the cancer question about cancer. We do curate HIV and cancer epitopes if they're in a paper that we would already be curating, like infectious disease paper, my influenza epitopes and HIV epitopes, we would capture both. The lag evolution that Bjorn mentioned, if they're from cancer patients, we would also include that.
Could you turn up your microphone just a little?
I don't know.
Or just stand closer to
Just
it.
stand closer. You just have to dirty it.
Thank you.
I'm afraid I'll get some kind of weird.
I'll see if I can get
Okay,
it.
yeah, I've been told I'm hard to hear. So all the data in IEDB is experimentally confirmed. We don't capture predictions. That's one of a big question we get from authors asking why their data is not in. If they publish a paper and file in predictions, we don't capture that. And then we also don't do review articles where the data was previously published somewhere else. It's a combination review article with a few new experiments. We would capture those new experiments. And then, importantly, we always capture negative data and supplemental data. A lot of our data comes out in supplements, and we do include the negative data so you can make conclusions when something is not recognized. And so each of the experiments in the IEDB shows the binding of an adaptive immune receptor to an epitope. So we have T-cell assays, B-cell assays, direct to binding assays, and MHC ligand evolution assays. And Carrie and Eve are going to go into great detail about all those assays. And then it always has to be epitope-specific data, but we do capture everything else that an epitope-specific receptor does. So if you have a known specificity antibody and it neutralizes whole virus, we can capture that too. If you have a known T-cell line that recognizes a specific epitope, if it binds to a whole protein or does something in an organism, we always capture all the experiments from the paper. So I think this also was a question someone asked. What's the minimum information for us when we're looking at a paper to include in the IEDB? So we made our cutoff for what's an epitope at 50 amino acids in length or 5,000 Daltons in length for the non-peptidics, just to have a way that we can consistently apply that across all papers that come out. If they test anything smaller than those sizes, we would call it an epitope and curate it. Most authors test something way smaller than that, but we still needed some sort of criteria. And then we do capture discontinuous residues also when they show that mutation or a substitution of a specific residue affects binding. We capture that as a discontinuous epitope. And then we also have non-peptidic epitopes, carbohydrates, lipids, chemicals, anything that the immune system is recognizing. And then the minimum information to curate is we must know the exact sequence for a peptide. If the authors just give start-stop positions, that's not enough to know exactly what was tested, so we'll contact the author if we need to. And then we need to know the host and the outcome. There are papers published where you never know whose antibody was being measured, so we can't capture that, but we do contact the author and try and capture it. And then sometimes you can't tell when you look at a figure if it's positive or negative. We can't tell if it's not called out in the text. Again, we have to ask the author or not curated at all if we can't tell. So we don't create the data, we capture experimental data from the literature. As Bjorn mentioned, those are the current numbers. We go to PubMed every 2 weeks, so we're always adding new data. And this is the process. So we do a complex PubMed query every 2 weeks to find things that are epitope related. We have a standing query that we run that is published. And we find all the epitope related literature that's come out since the last time we did it. And then we have an automated classifier that reads the abstracts and puts the articles in certain categories and the ones that we curate in the semester. So these are the categories we curate, but we do capture from the literature other things that are relevant, but we're not yet curating, like cancer specifically, but we do track what's in the literature and we're well aware of what is available. And then it goes to the curation team. So the curation team reads the whole paper and enters that information into a curation interface. If anyone's interested in that, we can show them during a break if you want to see how we enter the data. And then another curator reviews it, and when the two agree, we put it onto the website. And so we add about 15 new papers every week to the external website, and there's a lot of validation that happens during this process. And I think Bjorn's curve showed how sometimes we don't curate something. So about 70% of the articles that we get from PubMed turn out not to be curatable. And that can be because it's a prediction article, a review article, we can't tell what the host is, the author doesn't answer us, but we try really hard to curate everything. So the consistency and quality control measures. So we have a team of PhD level scientists at LGI. They're all around here today. And we also have one special chemist at KEBI, with chemical entities of biological interest, who has expertise in non-peptidic structures. So he draws those for us. And then we have a curation guidelines and peer review process that you can find on the website. There's a link to it. If you want to see what our curation guidelines are, they're really complicated and long, because the literature is so variable. We also use external immunological experts. So we're in an immunology institute right now, but we also contact different fields and authors to make sure we're doing things as best we can, and we're always asking for more help to make sure we're doing things well. And then the curation interface itself has validation when the data is being entered. It won't let you enter things that conflict, and there's a big check that's run with the whole curation once you're done with it that also checks again all the fields just to make sure they make sense. So Bjorn mentioned the submission community. These are primarily the Epitope Discovery contract holders that make submissions, but we will also take them for anyone who has a large data set or wants to submit even a small data set. And these are the current ones, as far as I know, of the new ones. So these are the authors, and then there's the subjects. There's mostly infectious disease, but also a lot of allergy, and it could in the future be any subject, discriminate. So the submission process is very similar to the curation process. The submitter will submit using tools. So we have an online thing where they can enter field by field, but most people choose to submit a large amount of data, so they use an Excel spreadsheet system. And then once it comes to us, we have a curator review it and work with the submitter to make sure everything looks like it should. And then again, we have automated validation that runs on those, the same validation that runs on the internally curated ones. And then we release the submission when the author tells us to. A lot of times they want us to hold it till the paper comes out. So the data structure. So the data structure, like Bjorn mentioned, is we have a lot of experiments that are all epitope specific. So we start with that reference that we get from PubMed, and we review it to see if it describes at least one epitope. Paper has one epitope, is almost curatable. It then has to have an experimental assay that shows an antibody binding or T-cell binding or image restriction. And if it shows that, then it gets continually processed. Most papers have multiple epitopes, and each epitope has multiple assays in most cases. And for the assays, there's a lot of fields that Carrie and Eva are going to go into in great detail. These are just some of the example of the fields that we capture: the host, whose immune system we're measuring, how did they become exposed, was there an immunization, were they ill, did they have an infection, was it a rabbit injected with protein, was it a mouse injected with peptide? We describe that because those differences cause a different reaction in the same assay. So an ELISA with immunogen virus might have a different results than an ELISA with immunogen peptide. So we capture all the level of detail for each of these experiments that you will see later. So I'm going to talk a little bit about how we organize the data in the IEDB. We like to use ontologies to do this, and the ontologies all exist outside of the IEDB for the most part as separate projects that provide standardized nomenclature, definitions, and synonyms, and a hierarchical relationship between terms for different data types. So for each of our IEDB fields, we see if there's an ontology that we can use to describe the data we enter rather than people typing something. Because each article will say the same thing a million different ways. We want to say it according to the approved nomenclature. This also helps us be more consistent and accurate for what's going into the database. It helps us find errors because often ontologies come with logical definitions that we can use to put validation in between fields. It makes curation easier because instead of typing, you're now selecting something from a defined list or a tree, and enhances the user experience as they can search using those hierarchies. So they can search in a high level or lower level based on the ontological relationships. And it also facilitates interoperability between our data in the back end and any other project that's using those same ontology terms. I mean, our data can mean the same thing, and you can use the data and put it together and draw new conclusions. So I'm going to describe a few of the ontologies and resources we use. One of them is a uniprot. That's how we build our protein trees. So most of the data in IEDB are peptidic epitopes, and they're described as a linear sequence that comes from a protein. And those proteins are described usually as isoforms, a specific hemagglutinin that will have a specific mutation that describes that epitope. But there's more than 350, maybe almost 400 different hemagglutinin molecules that we have curated data to over the years because it mutates so much. There's so many different sequences for the same protein. But we don't want you to search through 300 different hemagglutinins when you're trying to get flu information. So this hierarchical tree lets you search for all influenza proteins, all hemagglutinins, regardless of which isoform is used, and also processed fragments of that protein that occur in nature. And we get this information by using the UniProt resource. They provide all of this. We just map our data to it. And so we also get all the synonyms that come from UniProt, which can be really helpful when you're searching. Because there's so many different ways people write hemagglutinin. And then for the non-peptidic of this organism, the organism source of that protein that that epitope came from is organized by using NCBI taxonomy. And so the NCBI taxonomic tree is embedded into the curation interface and the search interface so that you can pick from everything that's in NCBI. But you can also search and sort by the higher levels. So you could select for all viral epitopes or all CMV epitopes or a very specific strain if that's what you wanted. And when you use the finders, you're searching the NCBI taxonomy. And again, this comes with all these great synonyms so that you can type the least amount and find what you're looking for. And then we also use Kebi. I mentioned we have a curator there who he reads the papers from the literature that we pull out, draws the structures and gives us the ID and gives it a name. And chemicals are quite complicated, so we really rely on their expertise, and they provide definitions and cross-references to other databases. And if the non-peptidic epitope has a non-peptidic source, we also use Kebi for that. Like the peptide from protein here, it's a small fragment of LPS was the epitope, and we capture both using Kebi. And for host, we also use NCBI taxonomy. All birth rates could potentially be studied. Whatever is published, we will capture that. So it could be a mouse or a rabbit, or it could be something more unusual like a llama or a shark. Whatever NCBI taxonomy will describe the host. And then for diseases, we use the disease ontology. And again, we get from them the definitions, the synonyms, and cross references to other databases, and it makes a tree that you can search. And then the assay type. So the assays are what IEDB is built all around. So we use the Ontology for Biomedical Investigation, it's OBI, and they provide this tree to us that's in the creation and the external websites. So our curators can find exactly what method was tested in literature, and you can search on higher levels. So you can search for all cytokine release assays, or you can search for all IL-2 assays, or you can search for a very specific one. So by using this OBI, we get a nice pretty tree that you can search on. And we embed this into the finder, which they will show you later. And for MHC restriction, we had to create our own ontology. So this is the one we've been the most involved with personally, because there wasn't a multi-species MHC ontology in existence. And we do this by modeling those as proteins with two chains that come together. And we also capture serotype and the genetic locus. So it depends on the detail available in the paper. Some authors might say this is a class 2 epitope, so we capture that. A different author may say that very same epitope has a very specific restriction like this EPA1. We capture whatever the author says in that paper, not what's known outside of the paper. So you may get a variety of restrictions, or an epitope may be restricted by multiples. But you can see how difficult this nomenclature might be if we had to type it out. So it's almost never written properly in a manuscript. But we want it, by entering it using the MHC restriction ontology, it's always the proper up-to-date nomenclature in the IEDB. And we include synonyms for anything we find in a paper for that MHC. And then the goal of the IEDB is to put all this data together in one place. So we have all the different manuscripts, and epitopes may be tested only once ever in history, or they may be tested many, many times. There are certain epitopes that have a lot of assays, so it becomes additive and informative. But you have to plan your query very well, because you'll get conflicting data if you don't, or there may actually just exist conflicting data from different authors. So that's why we go to so much trouble to have this workshop to talk about how to build your query, because you may see that the same epitope is positive and negative, and that may not make sense. But it could be that one is in a rabbit and one is a human, or it could be that one was a human with diabetes and one was a human that was healthy. So the exact how you build your query changes what the results are. But it can be very powerful because you'll see everything that's known and published to date, so you can draw your own conclusions. And then I think Bjorn showed one of these too. A different Timothy grass protein, showing how by putting all this data together, you can really see what's known about a protein. So this is amino acid 1 to the end of that protein, and then it shows you how many times an experiment was performed that we found in the literature, and how often it was positive, how often it was negative, and what was the response frequency among the people studied, if that was provided by the author. So you can see regions that have a lot of experiments that were positive. You can see regions that have a lot of experiments that were negative. So you can draw a conclusion of which parts of this protein may be relevant. But based on how you change your query, you may get a very different graph. So a biopsy mouse's immune response will look different from a human's, and antibody response will look different from a T cell response. So you can really map to which region of the protein is interesting to what you're studying. And then our goal is to accumulate as much data as possible. So this is the home page, and on one side we have a summary metrics that tells you currently how many epitopes, how many assays, how many references. And then down at the bottom right hand is the date it's updated. So once a week you should get new data. And then, as Karen mentioned, we always want help and feedback. We're here to serve you, so we want to know what you like and don't like and what you need. So there's lots of ways to get help at the IEDB. These little question marks link out to articles. There's help at the top and the bottom of every page. If you go to this Learn More link, you can get links to our curation manual. And the help and feedback exists as articles that we wrote in response to what that question mark was on the other page. And so every question mark has its own article. And you can also always email us and ask us questions. You can use this, or you can link online and just click that button. We try to get back to you as soon as possible. And you can go to our help page without having to ask a question. And then I'm trying to record these workshops and make them as online tutorials. Are we recording this now?
Yes.
I'm almost done. Then I want to let you know you can export the data. You can export all of the IEDB. We're free. We want you to use us. I'm pretty sure Carrie Nemo will show you how you can do a query and then export your results. But at the top and bottom of every results tab, you'll find ways to export the results of just your query. But you can also export the entire database. We have a page up at the top. There's a drop down. And if you go to the database export link, you get a page of all of the different formats. You can take all the data and do with it whatever you want. For writing tools or something, you might want to use the full set. And I'm mentioning this just because this is our newest export. We now have TCR and antibody sequences in the IEDB. It started relatively recently. And we have both nucleotide and protein sequences if the author provides them. We have CDR 1, 2, and 3 sequences if the author provides them, and we include gene usage. And if you export this table, you'll see every epitope that TCR recognized or every epitope that antibody recognized. And then we also have a 3D export that's been recently revamped. And it contains all of our 3D structures from the IEDB. If there's an antibody, an MHC, T cell that was shown by a ligand in a 3D experiment, we have that data captured. We include the PDB IDs of what's being bound, but also with the chains and full-length receptor and antigen sequences. And if you combine this information with the receptor export, you'll know a lot about the structures that are binding the epitopes. And that's it. Did I answer the question? Sorry.
That's all. I have one question. I have one question regarding non-peptidic epitopes. So are these, those epitopes, how do you find them? You know, because the sample that you put, it looked to me like a TLR ligand. So do you have data regarding?
Let's go back to that example just. So it has to be 5,000.
My question is whether this is recognized by an antibody, a TCR is recognized by the innate immunity.
And then the last one.
Innate immunity, so whether this is a kind of adjuvant or something.
Oh, no, so it has to be recognized by a T cell or an antibody.
Okay.
And
T cell, you know, that something is recognized by a T cell, or a B cell doesn't mean that it's recognized by the PCR or the TCR.
I don't know what you mean. So if it's
The
smaller
T cell
than,
receptor needs to be recognized by the T cell receptor.
Yes.
I mean, it's a question of definition, you know, but to define an epitope, you know, an antigen, it needs to be recognized by the, you know, a T cell receptor or a B cell receptor. So my question is whether these non-peptidic epitopes are recognized by, for example, TLR, like you said.
oh, I don't know. So we're only interested in TCR and antibody binding.
So if there is a compound that activates a T cell
Right.
as a mitogen or not through a T cell receptor, that would not be curated.
Okay.
It has to be an interaction
PCR
through the TCR or through the PCR.
Can you run the query just with that community?
It could very well be, let's say, if that is a TLR, it could be that someone made an antibody against it to be able to measure it, and that would be
You
curated.
remember?
Probably because the example is
Yeah, and in fact, for example, LPS, it's hugely used, antibodies against different LPS to serotype different bacteria, right? That is common. So that data would be curated.
Oh, I see.
Just go to the,
Firefox is open.
Okay.
Yeah, yeah. Seems like an identifier search.
Oh, yeah.
No, no, okay, 500.
I think you have to write this.
No, just 500.
I think you do.
No, I just tried it.
You did?
Yeah, 500. So these are all the assays in which this is defined. So you have like antigen inhibition, dissociation constant. So these are, I think these are.
Calorimetry, ELISA. So this particular one only had antibody hits. So there were no described T-cell responses to it, but there are an awful lot of antibody responses described to it. 27 references. And so when we get that, we do that query and get the papers, we read them to see if there's an experiment that shows a T-cell antibody binding. Then we send it to the curator, and he confirms its size is less than 5,000 Daltons. And then when it comes back to us in the manual curation, the paper is highly scrutinized for is this an epitope specific response.
I guess maybe along the same line. So does that mean you don't have any curation of NK or mate cell epitopes, or even like gamma does? It's all alpha beta centric.
It's not alpha beta centric.
Okay.
We have gamma delta T cells, and we have NK cells, and they often bind lipids. What?
NKT cell.
Yeah, NKT cells.
Right.
Yeah, he said mate. Yeah,
Just
we,
have-
yeah, it just has to appear in the literature, and we do
Okay.
include that. We have a receptors tab, which will be shown later, but you can see, we don't have a TCR here. So most of the data will be alpha beta. I don't think gamma delta should. Oh, yeah, no. We do have gamma delta, we have heavy light chain antibodies only, whatever is in the literature.
Okay.
There was one more.
So I may just miss it, but can you please explain what MHC ligand assay means?
So, one assay type is up here.
It's mass spec data.
So there's ligand dilution and there's binding. So there's all sorts of direct binding assays, like a fluorescent binding. Are you more interested in ligand dilution?
Actually, I want to ask a question regarding ligand evolution.
Okay, but I wanted to make,
Just in general.
So MHC ligand is our aggregate of MHC binding and MHC ligand dilution. So it's just a parent term, so that both types of that exist.
so it can be isolated free MHC binding, it can be cell-bound MHC binding, it can be a variety of assay types. Flow cytometry, any experiment type that we encounter in literature, we get added to OB and then use it. But for ligand dilution, it's mostly,
Right, so the bulk of the data, even though all these different assays that she's mentioning are curated, the bulk of MHC binding is when you have purified MHC tested with synthetic peptides to measure the interaction, and likewise, the bulk of the ligand dilution, it's data determined eluting of peptide and then sequencing by mass spec, which is now generating large amounts of data. Does that answer your question?
oh, yeah.
So how do you get the mass spec data? Is it the elution, MHC elution then?
You'll be shown
Oh, okay.
many queries, but, so, as I mentioned, the MHC is the superfamily for binding and dilution. If you just pick
Like
dilution
that.
only, you'll get just dilution data. If
Okay.
you pick binding, you'll get just binding data.
Okay.
If you
Thank you.
pick parent, you'll get all the data. And in general, I think we usually advise to make a more generic search and then narrow it once you see how many results.
Okay.
And you can also limit it to certain MHCs, so we do have that as a search parameter. If you're interested in specific MHCs, you'll get a lot narrowed data set.
Can you clarify what defines linear epitope? Because you said less or equal 50 amino acid, but wouldn't, like, for example, MSC1 is between 8 to 10 amino acid.
Right.
But
So
why you have more?
Most authors test pretty small peptides, but some, there's, especially in older papers, sometimes they'll make 25 mers, 30 mers, 40 mers. If that's all that's published, we capture it because otherwise there might be no data on that protein. But
And then there's a difference between, we don't think that that's the minimal epitope. You call that an epitope
evidence
containing
codes.
region. So the 50 residues are a threshold that an epitope containing region has to be narrowed down to at least 50 residues. So that's.
do you guys talk about
And
evidence
the-
codes?
And I think she might be interested in the range that we have for class 1 versus class 2.
Yeah. So we have evidence codes for how narrowly defined the epitope is. We have epitope containing regions for large regions that were tested and not further refined. And we have exact epitope for regions that are small enough based on the MHC to be more fully defined as the epitope. And because some authors might only test large peptides and another author might do the refinement, we capture all of it. And that's why on that graph, you see it all, it's all put together, whether someone tested a long peptide or a short peptide, it all accumulates along the length of that peptide, protein.
So do you have 3D structures for these linear epitopes? MHC, class
3D?
2 bound to the peptides? Do
3D?
you annotate those or not?
I don't understand.
The 3D PDB structures?
Yes. You can click for all 3D structures, but Carrie says you're going to see that later. But if it's in PDB and it's an antibody, a TCR, or a MHC binding to peptide, we have those in the IEDB. And we do that query alongside our regular query every 2 weeks. And this little icon, click on that, it brings up. They're going to show you that.
So this is empirically driven, that you get your epitopes from the literature. But have you used, for instance, for MHC binding, have you used all of those to come up with predictive algorithms for binding for specific proteins?
Yeah. Yes, so tomorrow will be all about the prediction tools. There's a whole side of the database that is the prediction section. And there'll be talks about each of the tools, and those tools do use our data sets to help train them. And we hope other people not even affiliated with us, we hope they're also using our data for those purposes.
I actually have a very interesting, at least to me in our work, MHC molecule I couldn't find in your database. We can talk about it
Yeah,
later.
tell me
when
more.
I tried to look at my data. It may be because it hasn't been reported in the literature.
Yeah.
It's regarding the curation. So how do you take care of the publications which are like not in the public domain when you search from the government?
You mean not available for free?
Yes.
Because we're at an institute, we have UCSD access. But if we can't access it, that is a reason some papers become uncuratable. If they're not English language, or if we can't get a copy of that paper.
Another question is, do you incorporate like natural language processing while curating the stuff from the applications?
Natural ligand processing? Is that what
Yeah, it's like using the machine learning to extract the keywords from the text, and which could complement your ongoing.
Okay. So we only use automation for screening the abstracts to categorize them. We then manually do the next step. A problem for us has been that a lot of the information we're capturing is so detailed. Like if you found that the word human was mentioned in a paper by machine, you don't know if that's human is the host or human is the source of the protein, or if it's not even related to the curatable aspects of that paper.
But that can be, customized, keeping all the phrases into taking into the account.
In the value creation world, there's a lot of efforts in trying to do that. If you have, we can talk more if you have good ideas.
I was follow up on that is, are you using an off-the-shelf classifier for the documents, or is it custom trained on like a sample of the abstracts?
Custom trained and retrained every year, I think.
Fantastic.
Because custom built, custom trained, and
Awesome.
continually retrained.